All right, well, thank you to the organizers and the KCA for inviting me to come and tell you a little bit about my science. So my name is Ed Resnick. I'm a computational biologist at Memorial Sloan Kettering, and today I'm going to tell you about metabolism in RCC and in the RCC tumor microenvironment. So I have no disclosures. So as I said, I'm a computational biologist, and my lab is primarily interested in studying cancer metabolism. And today I'm going to tell you about one prong of our research program, which really relates to using high throughput measurements of hundreds of metabolites in tumor specimens, which we call metabolomics, to understand tumor physiology, to understand the progression of disease to a more aggressive state, and also to understand therapeutic vulnerabilities and opportunities, and how this is applied to kidney cancer. So my interest in this really started about 7 years ago when I began working with two colleagues at Memorial, Ari Hakimi and James Shea, who had generated a really exciting data set where they had metabolomically profiled 140 primary ccRCC specimens and their adjacent normal tissues. So when they did this, they measured the abundance of about 500 or so metabolites. And the goal of the study was really to understand what the metabolic differences were in tumors and how those were contributing to malignancy. So I don't want to belabor this study too much. You should feel free to go out and read it. It came out many years ago. But I do want to draw out some interesting findings that we made that will kind of come into play later on in the talk. So one of the first things that we notice is that kidney tumors are really quite different from adjacent normal kidney metabolically. And those differences are really focally concentrated in pathways. So for example, many gluconeogenic sugars and glycolytic intermediates are elevated in kidney tumors relative to normal, and that probably speaks to a higher rate of glycolysis in tumor cells. And sort of unexpectedly, we found that the abundance of almost every amino acid, with a few important exceptions, was uniformly depleted in kidney tumors relative to adjacent normal tissue. Now, what was more interesting was that when you started comparing aggressive tumors, so let's say high-stage tumors, to low-stage tumors, sort of surprisingly, we noticed that many metabolites seem to show very profound changes in abundance as the tumors became more and more aggressive. Now, after years of looking at lots of different metabolomic data from many different cancer types, I can tell you that this is not the kind of finding that you see in every cancer type. In fact, it's, I would say, relatively unique, or it's most prominent in kidney cancer. It really does seem as if the progression to advanced disease is sort of centrally and critically associated with changes to metabolism. Now, one of the sort of unexpected findings from our original study was that when we did a broad principal components analysis of the 140 tumors that we were studying, 2 of them appeared to be outliers. And when we went to our pathology colleagues and asked them to tell us if there was something strange about these tumors, they told us that yes, indeed there was. They were misclassified as clear cell tumors, and in fact, they were clear cell papillary RCC. So for those of you who are not familiar with clear cell papillary, it's a relatively rare subtype of RCC. Patients typically have an indolent course. It's probably the 4th or 5th most common type of RCC, but because of its relative rarity compared to other kinds of cancer, there's not much known about the molecular drivers of clear cell papillary. So like many of our colleagues at Sloan Kettering, when we found out that these tumors have a very interesting metabolic phenotype, we decided to sequence the hell out of these tumors to learn about what their molecular and genetic drivers might be. So we conducted a variety of whole genome and whole exome sequencing, and the outcome of that was that we learned that these tumors are remarkably quiet. They have absolutely no copy number aberrations. They are completely diploid and fully heterozygous, and they have an extremely low mutation burden. So surprisingly, we found not a single gene that was mutated more than once in this disease. So it seems like, at least from a genomic perspective, there are not many strong genetic drivers in clear cell papillary. And that is probably consistent with the fact that they are indolent. What is more interesting molecularly about these tumors is that they have a really profound metabolic phenotype. And that phenotype is derived from primarily from one metabolite that is elevated, let's say between 50 and 100-fold in these tumors, and that metabolite is sorbitol. So let me tell you a little bit about what sorbitol is. It's a sugar alcohol, and it's derived from glucose. So when you catabolize glucose traditionally and primarily in most cells, the way it's catabolized is through glycolysis. There are alternative ways to catabolize glucose, and one of them is through something called the polyol pathway, which passes through sorbitol and fructose and eventually merges back with glycolysis. Now, there are physiological reasons to push flux through the sorbitol pathway, and I just want to draw your eye to one important consequence of doing so, which is that in order to make sorbitol and then get rid of it, you need to pull an electron off of one very important redox cofactor, NADPH, and you need to put it on a different redox cofactor, which is NAD, to make NADH. And that observation became very hypothesis generating for us. We basically posited that one reason you may see an aberrant accumulation of sorbitol is if the forward reaction from sorbitol to fructose is really unfavorable. And one reason that might happen is if you have a lot of NADH accumulating inside of the cell. And that could happen if there is a disruption to the integrity of the mitochondrial genome. So let me tell you a little bit about mtDNA, or the mitochondrial genome. So it is a very small circular genome present in hundreds to thousands of copies in each one of your cells. And it only encodes 13 proteins, but those 13 proteins are absolutely essential for mitochondrial oxidative phosphorylation and respiration. Most importantly, many of those proteins encode subunits of complex 1, which is the primary cellular pathway for oxidizing NADH to get rid of, to regenerate NAD. So the premise here now is that if there is an insult to the mitochondrial genome, complex 1 doesn't operate, and then you get accumulation of NADH. So when you start investigating the mitochondrial genome of clear cell papillary, you don't find mutations. And the reason you don't find mutations is, in fact, because clear cell papillary is exquisitely depleted of mtDNA. So clear cell tumors themselves are very, very, very depleted of mtDNA. And even with respect to clear cell tumors, clear cell papillary distinguishes itself. So in both whole genome and whole exome sequencing, these tumors are depleted more than 10-fold relative to adjacent normal tissue. If you go to the TCGA, there are 5 clear cell papillary cases there. They're also mtDNA depleted to the same extent. That depletion translates to a drop in mtDNA encoded RNA, and it translates to protein levels. So if you do immunohistochemistry for mtDNA encoded proteins in clear cell papillary, you see a huge drop in protein expression that you don't see in ccRCC or in normal tissue. And the same is true just generically for mitochondrial mass. So the mitochondria in these tumors are highly disrupted. So all of this kind of points to a model that we have for clear cell papillary, which is that it has this very unique metabolic phenotype, the accumulation of sorbitol, and that is driven probably by a disruption to NADH to NAD ratio in the cell that is caused by an inability for complex 1 to function because there's really not much mtDNA in the cell. Now, the dangling question here is why there is no mtDNA, or why there is so little mtDNA in these cells. And that is something that we still do not understand and we're still working on. So in the course of doing all of this metabolic analysis, I've constantly had this question ringing in my mind, which is where the metabolite signal is coming from. And what I mean by that is that when I think we can all appreciate that kidney tumors are not purely composed of malignant cells. They're obviously composed of non-malignant stromal cells, immune cells, and the tumor cells that we intend to study when we do a lot of this profiling. So obviously, when you do bulk metabolomic profiling, you are picking up a heterogeneous mixture of metabolites that are derived from each one of these cell populations. Now, this becomes interesting, that kind of intuition becomes interesting when you think about how the composition of the tumor microenvironment might influence bulk metabolomic measurements. And what got me really intrigued was an observation that my colleague Ari Hakimi made, where he took TCGA data and stratified tumors by whether they were highly, modestly, or not at all T-cell enriched. And what you notice is that T-cell enriched tumors have much lower expression of mitochondrial genes and metabolic genes in general. And that really suggests to us that there is potentially a relationship between the composition of the tumor microenvironment and the metabolic phenotype of the bulk tumor. So in order to study that, my lab has been assembling something that we call the multimodal metabolic map. So what this is a collection of hundreds of tumor specimens or adjacent normal specimens from a variety of diseases for which we have paired metabolomic measurements and transcriptomic measurements, either by RNA sequencing or by microarray. So much of this is public data, and in fact, if you have such data and you want to contribute to the effort, please feel free to reach out to me. So the benefit of one of the benefits of the multimodal metabolic map is that we can infer TME composition. We can infer the cell populations which compose the TME using immune signatures and applying those signatures to RNA sequencing data. And that enables us to study TME metabolite interactions. So focusing on kidney cancer, one of the first things we wanted to study was not necessarily how the microenvironment related to metabolism, but how gene expression itself related to metabolism. And one of the most intriguing observations we made right off the bat is that there are pairs of metabolites and genes where these genes encode enzymes that are recurrently correlated across the 4 data sets of kidney cancer data we have in the MMM. Now, the other special thing about these metabolite gene pairs is that they are functionally proximal to each other. In other words, that gene is either the direct producer or consumer of that metabolite. And there are 2 really critical implications of this. One is that potentially what we're looking at are rate-limiting determinants of metabolite abundance. But more importantly, I think that what is really valuable about this is that these genes represent surrogates for metabolite levels. And what you can do now is go to public RNA sequencing data and do in silico metabolomics where you infer metabolite levels using these gene signatures. And that's really a way of democratizing metabolomic data analysis using widely available and public RNA sequencing data. So that's something that we're pursuing right now. Now, with regard to the microenvironment, when you use established and validated RNA signatures to infer the cellular composition of the tumor microenvironment, one of the first things which really strikes out is that there are a subset of metabolites which are strongly and recurrently associated with the abundance of specific cell populations in the microenvironment. Now, we're still trying to understand what that means. It could be that those cell populations really produce or consume or just have at high abundance that metabolite of interest, or it could be that the presence of that cell population fundamentally changes the metabolism of different kinds of cells in the microenvironment as a whole. So that's really an open question that we're trying to figure out right now. So with that, I just want to thank the wonderful students and collaborators and mentors who helped me conduct all of this research. I want to thank my funders, and I just want to advertise that my group is looking for eccentric and excited computational scientists to lead this sort of multimodal metabolic analysis going forward in kidney cancer. So thank you.